Hi everyone, welcome to topic 1, and this is on pharmacodynamics. This topic is split into 3 parts, and in this very first part we're going to talk about general principles of pharmacodynamics. Now here are all of your lecture objectives for this part of the topic. On each slide though, across the top, we'll have the specific learning objective that relates to that content on the slide. So pharmacodynamics then is how the drug affects the body. So here we're really talking about the mechanism of action of those drug molecules. So whether it's a Nurofen that you've taken for inflammation, whether it's beta blocker that you might have to take for hypertension, or whether it's an inhaler you take for asthma, the pharmacodynamics of all those drugs describes how those drug molecules, come into contact with target cells to have a particular effect. In contrast, then, pharmacokinetics is all about how the body processes drugs. Now, as I mentioned in the introduction, the most common way that we do take drugs is in oral or tablet form. There are other modes of administration that we'll talk about when we get to the pharmacokinetics topic. But pharmacokinetics then describes how the body absorbs those drug molecules, how it distributes those molecules to target tissues, how it metabolizes those drug molecules, and eventually permanently excretes those drug molecules from the body. Now, there is a massive overlap between pharmacodynamics and pharmacokinetics. For example, the pharmacokinetics will affect drug action by determining which tissues the drug gets exposed to. Elements of pharmacokinetics are all dependent on our genetics. For example, metabolism of some drug molecules differs from person to person. And we'll get onto that when we get to the pharmacokinetics topic. But because, there is such an overlap between pharmacodynamics and pharmacokinetics, it is hard to talk about them individually, but we are going to do that over the next couple of weeks and then bring it all together. So what we're going to do now is look at some general principles of how drugs act. These next couple of slides are a little bit wordy, but I really need everybody to concentrate so we get this foundational stuff really right before we move on to some of the more complex aspects of this topic. All right, remember that drug molecules need to bind to cells, right? And remember that drugs are chemicals, okay? So a drug or a chemical needs to bind to a constituent or a component of the body to initiate a pharmacological response. Now, drug molecules then must exert some sort of chemical influence on one or more constituents or components of cells in order to produce a pharmacological response. And this means that drug molecules, or these chemicals, must get so close to these constituent cellular molecules that there's a chemical interaction between the two in such a way that the constituent cellular molecules is altered, their function is altered. Okay, so as I said, that's a bit wordy. What I'm going to do now is try to explain it in a far easier way using Lego, because remember, everything is easier with Lego. But I'm going to need you to stretch your mind here a little bit. Okay, here we have a little setup here where we have this grey flat piece of Lego, which is representing the extracellular side of the plasma membrane. We have this door here in our plasma membrane. I don't think you can quite see it, but that's resting on another piece of flat Lego, which is representing the intracellular side of the plasma membrane. Okay, we have this red flat bit here, which is some sort of receptor on the surface of the plasma membrane. And we've got this door here, which kind of represents an ion channel that's, crossing either, both sides of the plasma membrane. All right, so keep that in mind. Now we've got a drug molecule here in brown and yellow. And yes, I'm aware this is not drawn to size. So what happens in this instance is this drug molecule binds to a part of that receptor on the plasma membrane. And what do you think is going to happen? Well, we've got our door representing our ion channel. it's going to cause the ion channel to open. And we know when ion channels open, there's either going to be an influx or an outflux of ions from a cell, and that causes a cellular response. When the drug molecule then moves away or dissociates, the ion channel closes again. And it can then be, stimulated or activated again if a drug molecule binds to that receptor again. So leading on from this, in terms of drug action, it does require, firstly, the non-uniform distribution of the drug molecule within the body or within a tissue. So remember, uniform distribution is where, in this case, the drug molecules are evenly dispersed within the body or a tissue. But for example, if you want to design a drug that acts on brain tissue, it needs to be able to cross the blood-brain barrier to act and, to accumulate and act on brain tissue. Secondly, drug molecules must be bound to particular constituents of cells and tissues in order to produce an effect. And that's what you're looking at here in this little Lego animation that I made. The drug binds to this surface receptor, opening the door or the ion channel. And finally, it requires an understanding of the nature of the binding sites and how the interaction between the drug molecules and the binding sites leads to a physiological action. Now, you should be very familiar with binding sites and ligands and the specificity that exists between the binding site and the ligand. Remember, we did that in previous physiology subjects. And we talked about things like hormones, and hormones being signaling molecules that bind to receptors, and that binding then stimulates a series of reactions to, stimulate a cellular response. So same thing here. A drug molecule can bind to a receptor, and the effect of that binding can be either to enhance the effect of the endogenous ligand, so it mimics the effect of the endogenous ligand, or it can be used to inhibit the binding of the endogenous ligand. And that's what we're going to talk about now in the next few slides in terms of agonist drugs and antagonist drugs. Okay, some important terms here that we need you to know about, and they are the terms selectivity, specificity, and affinity. Now, when designing a drug, to have a particular effect in treating a disease, so for it to be used as a therapeutic tool, it must act selectively on particular cells and tissues. Now, selectivity refers to the number of molecular targets that drug molecules interact with. And it must also show a high degree of binding site specificity. And this refers to, the number of effects that a particular drug has. It must also display affinity. And affinity refers to the strength of the interaction between the drug and its molecular target. Okay, so you want a drug to have a high affinity, right? It's also known as avidity, and it's important, in terms of the length of time and that a drug binds to a target, and also the types of bonding that occurs between the drug target and the drug molecules. Now, proteins that function as drug targets generally show a high degree of ligand specificity. That is, they bind only molecules of a precise type and shape. Specificity is reciprocal. There is binding site specificity, right? So specific drugs only bind to certain targets. And there's also ligand specificity as well. Individual targets will only recognize certain drugs. This is kind of shown here in this figure here. You can see the binding site specificity and the ligand specificity as well. This is a schematic representation of a G protein coupled receptor by drugs. You don't need to memorize all of the details of this slide at this stage, but you might need to go into some of this stuff when you're doing your assignment. But all I want you to notice here is we've got our ligand and our binding site specificity. We get that activation there of that G protein coupled receptor that triggers a whole load of downstream reactions to occur, and then the functional response might be smooth muscle relaxation, or it might be a modulation of an ion channel, or it might be smooth muscle contraction. Okay, now if it were possible to design a drug that was 100% specific, it would mean it would have one singular molecular target, it would act at a singular site, and it would only have one effect. And that is pretty much impossible to do. And we've given you an example here of the histamine antagonists. Now, it doesn't matter if you don't know what an antagonist is at the minute, but it is also known as a blocker. So it's a drug that blocks the binding of the endogenous ligand. In this case, it's histamine to the histamine receptor. So histamine antagonists are also called antihistamines, right? Now, you might take an antihistamine if you've got sort of an allergic reaction that causes, runny eyes and runny nose and sneezing, okay? And you get relief from that. But sometimes you might also feel a bit drowsy, or you might actually fall asleep. And that's because there are, histamine receptors in the central nervous system, as well as on immune cells, which is why you take it for the allergic reaction, also in your gut and elsewhere too. So when you buy an antihistamine and you turn it over and read the back, sometimes it says this medication may cause drowsiness, do not operate a forklift, okay? So you really need to, read the back of the box if you are intending to drive a forklift or more commonly a car. Okay, so just to point out here that, no drugs are completely specific. And side effects then are often due to the presence of recognition molecules in other parts of the body than the target tissue. Now, side effects might be beneficial. We mostly talk about them in terms of unwanted effects or bad effects. And sometimes, side effects can be very serious. They can be even sort of fatal. And when you open up, a box of tablets or whatever, and you take out the information pamphlet, you'll see a whole plethora of side effects. It doesn't mean that everybody is going to get these side effects. Some of them might be common, some of them might be very rare, but the makers of the drug, have to mention though that it might, it is possible to get one of these side effects, or else they'll be liable. Okay, here we'll go back to these histamine antagonists. Sometimes you might have a kid who's covered in mozzie bites and they're scratching all the time and they're not sleeping. So you might actually decide to give them an antihistamine that also crosses the blood-brain barrier and binds to the histamine receptor in the central nervous system to make them able to sleep throughout the night. But as I mentioned, some side effects are often unwanted, and they can be toxic, and in particular toxicity to the liver and the kidneys. Two other really important terms in relation to drugs are the terms potency and efficacy. I'm going to start with efficacy, and it is the ability of a drug to produce its biological response. So I guess efficacy is the maximal response that a particular drug can produce. It's really important clinically. Some drugs display the same efficacy, whereas other drugs have different efficacies. So when we're talking about efficacy, we're talking about how big an effect a particular drug can have. Now, we'll look at this figure in a minute, but we'll talk here about potency now. And you often hear that, oh, that's a very potent drug or whatever. Potency is how much of the drug is required to produce a response. Now, it's determined by the affinity of the drug for the target and also the drug efficacy, so the ability of the drug to produce its biological effect. And it's compared by considering the EC50, which is a concentration at which the drug produces 50% of its maximal response. And I really want to stress of its maximal response, so it's individual to the particular drug. Okay, let's go over here and spend a couple of minutes on this figure, right? This is a theoretical concentration response curve, also known as a dose response curve. And these are three different drugs. So drug A, drug B, and drug C. On the y-axis here, you can see the percent maximal response of a tissue. It goes up to 100. And then on the x-axis across here, you have increasing drug concentration. And it doesn't matter what the units are of these drugs, in terms of concentration at this stage, right? You can see that this is a log scale here. Let's just say, for convenience sake, these three drugs are bronchodilators, okay? And a bronchodilator relaxes, the smooth muscle in those little tubes called bronchioles in the respiratory system. So these drugs are reversing the bronchoconstriction that occurs in an asthmatic episode. So most people who have asthma will carry one or more of these little inhaler bronchodilators with them at all times. What I want you to see here, and we will get onto this a bit more later, there is a subtherapeutic effect here. So this is the concentration of drug that's not giving you the therapeutic effect. Then we go to this wide sort of range here of, a tissue kind of response that you're getting the therapeutic effect. And then we have here, increased risk of adverse reactions occurring up here. Okay. Now, what I want you to notice straight away from these three sigmoidal graphs is that these are agonist drugs because they are producing an effect. Now, I will get on to agonists and antagonists in a minute. And you can see all three of them are producing the maximal tissue response. What we're interested in here at the minute is looking at potency, okay? So a very highly potent drug can be used at a low concentration. So clearly this drug A, which is producing maximal dilation, bronchodilation, is doing it at a very low concentration. Drug B is just as good at producing the bronchodilation. It just needs, a higher dose, so it's less potent than drug A. And drug C then is the least potent. It's still producing the maximal effect, but you need way more of the drug for it to do so. So A is 3 times more potent than B, which is 5 times more potent than C. Have drugs with the same potency, but different efficacies. So let's keep with the bronchodilators here for this example too. You can see that the purple curve here is producing the maximal bronchodilation, okay? But we've got drug B here, which it doesn't matter how much you increase the drug concentration down the bottom, you're never going to get it to produce the same response, same amount of bronchodilation as the purple curve. And then C is only producing its maximal response of 40% here. Okay, so I'm sure you can all see that these three drugs have different efficacies. Now I'm going to tell you that they have the same potency, right? And you might be wondering, why is that? Well, remember that the potency is related to the EC50, and that's the concentration of drug that produces 50% of its maximal response. So if the maximal response of drug A is 100%, 50% of that, if you draw a line across here where it intersects with the purple curve and come down to the x-axis, you get a drug concentration of 10, okay? Now, we can see for the green curve, it's 80%. So 50% of 80 is 40. So again, we draw a line across to the green curve, bring it down, and we get 10 again. And this little one down here only can reach 40%. 50% of 40 is 20. So again, we get a drug concentration of 10. So in this case, you're looking at these dose response curves or these concentration response curves for three drugs with the same potency but different efficacies. Now, I've just put a note here just to remind us that higher potency does not necessarily mean more side effects. Remember, potency relates to the amount of a drug that you need to give to have an effect. So theoretically, if you're using the drug at the correct dosage, you shouldn't be generating more side effects. But obviously, as the dose of a drug is increased and increased, you're introducing more chemical molecules, into the body, and drugs tend to become sort of less selective and less specific and sort of start binding to different, molecular targets. And that's when you can get more side effects. And remember as well, I'm not sure if I've mentioned this already, but, not everybody gets a side effect that's the same as another person's side effect from a drug, or they might not get a side effect at all. So while you're listening to me, just think of, any kind of common side effects that you may have sort of experienced in taking an even over-the-counter drug. Okay, so very briefly, I am just going to introduce the 4 major types of drug targets, because in the next recording, we're going to be looking at that interaction between drug molecules and receptors more closely in terms of agonists and antagonistic drugs. Okay, so we have receptors. The example given here is the beta-2 adrenoceptor or adrenergic receptor in the lungs. It's a receptor agonist and it causes bronchodilation, right? Enzymes can also be a protein drug target in this table. The example there is the HMG-CoA reductase in the liver. The drug is inhibiting that enzyme, and that can cause, a reduction in plasma cholesterol concentration. Transporters or carrier molecules, and these are carrier molecules for sugars and amino acids. The example given in your textbook is the serotonin transporter in the brain. It inhibits, the mechanism of drug action is to inhibit this transporter, and the response to the drug then is a reduction in depressive symptoms. So it's used as an antidepressant, right? And then we have ion channels, lots of different sodium, potassium, chloride, calcium channels. The example here are calcium channels in blood vessels. The mechanism of drug action is to block the channel, and here the response to the drug then is decreased vasoconstriction. So this might be given to somebody with hypertension. And just focusing in on receptors here, they are recognition molecules for chemical mediators. So these are molecules through which soluble physiological mediators such as hormones, neurotransmitters, and inflammatory mediators produce their effects. Example is the acetylcholine receptor, which you're looking at here at the neuromuscular junction. There's also cytokine receptors, steroid receptors, and growth hormone receptors. So, these are all receptors for physiological mediators, but they can also be the target for drugs as well. Okay, I'm going to stop here, and when I come back, we're going to be focusing in a little bit more on the drug receptor interaction.